Hi, good morning or good afternoon to everyone. Thanks so much for joining us today in our webinar titled AptaFluor Detection Platform, a Homogeneous Universal Methyltransferase Assay based on a microbial ribose switch. Some of you probably are already familiar with BellBrook Labs. For those who are not, a very brief introduction. We are a small biotech company based in Madison, Wisconsin. And we have been selling reagents for high throughput screening assays for a little more than 15 years now. And we take a lot of pride in the kind of reagents we sell. It's branded under Transcreener assays. And these are antibody-based assays that offers direct detection of nucleotide products from either kinase reactions, GTPases, etc. Of course, that's not the topic of today's webinar. Today's webinar would be about AptaFluor Detection Platform. This is one of the newer, this is the new platform that we are launching, or we just launched it commercially. And I'm going to be talking about this assay format today. Just a couple of housekeeping notes. So should you have any questions, please feel free to type them in the little box that says questions, and we would try to get to the questions by at the end of this webinar. Also, if for some reason I'm unable to address your questions, please feel free to email us your questions and we'd be happy to answer them. So with that, let's get started. Okay, so ribose switches were discovered in 2002 by Ron Breaker at Yale, and he discovered them as RNA-based intracellular sensors for vitamin derivatives. Excuse me. So during the last decade, several ribose switches have been identified, most of them in bacteria, but there have also been ribose switches that have been identified in plants and in fungi, and they bind to a range of biomolecules, some like nucleotides, either amino acids or even ions, and exert regulatory control on transcription, translation, splicing, and mRNA stability. So this image that I borrowed from a review article that appeared in Nature shows how a ribose switch works. So a ribose switch primarily has two regions. One is the aptamer domain, and one is the expression platform. So in this first figure that you see, -M, meaning no metabolite, is bound to the ribose switch. There's a switching sequence. It takes up an anti-termination loop. It gets the confirmation of an anti-termination loop and lets the transcription happen in the mRNA. So this is in the absence of the metabolite. Now, when the metabolite binds to the ribose switch, as you can see, there's a change in conformation, and instead of the anti-terminator loop, the switching sequence adopts a termination loop and thereby terminates the transcription of the mRNA. Now you can also see that the two loops in the aptamer domain also comes closer together. So this is how typically most ribose switches perform. So the ligand binds and there is a conformation change that happens. Now, a little bit about methyltransferase assays. Assay methods for methyltransferases depends on two things. One is either detection of the methylated product that's shown here, or detection of SAH, that's the invariant product that's formed in the methyltransferase assay. Now, methylated product, the most quantitative and the most reliable method would be a radioassay. However, there is an associated regulatory and disposal cost associated with any radioassay, and that makes it a liability for high throughput screening assays. So Detection of methylated products are being done using other methods like antibody-based or protection assays. But the problem with these kind of methods is that A, you need to have different antibodies for different methylation products, and B, sometimes you have either dimethylated product or trimethylated product, so a single antibody cannot detect all those different methylated products. So the best method, therefore, for a methyltransferase assay would therefore be to detect SAH. Now, there are so many coupled assays out there that detects SAH, and BellBrook Lab also has one of the coupled assay for detection of SAH that's sold under Epigenetic assay. Now, these assays are very good, but one of the problem, A, it's a coupled assay, so it's not direct. That's one of the issues, because you have to go back and deconvolute your assay if you're running a huge screen. B, the other issue with a coupled assay is since it's coupled and it's not direct, it doesn't give the sensitivity that is required for some methyltransferases. So there are some methyltransferases that have very low Km for SAM, and we heard from our customers when we launched our Epigenetic assay that, hey, it's good, but it's not sensitive enough. So we wanted to address that, and our target was to develop a direct assay, and B, to make it very sensitive, so it really solves an issue. Today I'm going to be talking about how BellBrook Lab has used aptamer-based assay to develop a methyltransferase assay. Now, the AptaFluor Methyltransferase Assay relies on highly specific RNA aptamer or ribose switch that I showed earlier. Now, this was evolved in nature. It occurs naturally in bacteria as a SAH responsive sensor. It binds SAH with really low nanomolar affinity and about 20 to 10 nanomolar, as you can see here. And with exquisite selectivity. Now, that's really important when you want to develop a good assay. The selectivity is the primary importance. So we saw that between SAH and SAM or adenosine, there was a really good selectivity. SAM did not bind until you reached about a micromolar amount, and SAH had nanomolar affinity. So these properties made it a perfect affinity reagent for a methyltransferase assay, because a specific detection of SAH in the presence of excess SAM is what is required to measure at initial velocity conditions. And so the next challenge for us was to figure out how are we going to transduce this into a robust assay. Now, so initially we started with the intact ribose switch, and it posed several challenges to us. And we were not able to reach the sensitivity that we had targeted. So we got around this problem by splitting the aptamer into two halves. So as you can see in this graphic here, one half, the yellow one that's shown, is attached to far red fluorophore, and the other half shown here is attached to biotin, which is further conjugated to streptavidin terbium. So we just show here as terbium in this graphic. So what happens is, when the methyltransferase, as the reaction progresses and as SAH is formed, the SAH induces these two halves, the two parts or the two split parts to come together and recreate an intact aptamer, and we could then use it to measure it using TR-FRET. That's what is shown here. We also have some data with FP, although we are just launching the TR-FRET assay at this point. The Dilight 650 is the far red fluorophore that we use, and far red fluorophores are known to minimize interference from fluorescent compounds and light scattering. So that helps in minimizing, false positives in the assay. The assay itself is can be run in two steps. So the first step is your enzyme reaction that you would typically run, where you add your substrate. Basically, you can run this assay with any substrate. It's a universal assay. So nucleosome or core histone proteins or peptides, anything. And then you add your SAM and your enzyme, and the reaction progresses. And the next step is where you quench the enzyme reactions, and then you add the detection reagents. You incubate it for a certain period of time and read the plate. So it's really simple assay. There are only 2 additions that's involved. And you have the ability to also perhaps run it in kinetic mode if you're running the assay using peptide as your substrate. You can run, you do not need to quench, you can also run it. We require that the assay requires that you need to quench if you're running it with histone proteins and nucleosomes. So this flowchart, I guess is what it's called. So this flowchart walks you through the assay setup. So you start, as I said, you start with your 10 microliter enzyme reaction. Of course, there's flexibility. If you want to make it even more sensitive, you can incubate a 15 microliter enzyme reaction. So we leave it completely to the choice of the investigator to formulate the experiment as per their choice, but this is just how it is, how we suggest that you do in the tech manual. So you run your enzyme reaction, you incubate at whatever time and temperature that's specific to that target that you're working with. And then you add your 5 microliter stock reagent. You incubate 10 minutes. The key here is to bring the temperature, especially if you're running your methyltransferase reaction at 37 degrees. We would like you to bring it to room temperature, so you can even incubate it longer just so it reaches room temperature. And that's imperative. It's very important because we want the 2 pieces of RNA to come together to anneal, and the best, optimal annealing temperature for those 2 pieces when SAH is present is it's at room temperature between 23 and 26 degrees. So that's really important for a good assay development. And then you add 5 microliter of SAH detection mixture. So as I mentioned, I'm going to just go back one bit. So as I mentioned in my previous slide here, so when you receive a kit from us, you will have 2 components. One is the part 1, we call it piece 1, sorry, piece 1, P1, which is conjugated to biotin, and this is piece 2, which is P2, conjugated to dilite 650. So we just, before you make your detection reagents, we just ask you to conjugate the biotin-related piece of RNA with streptavidin terbium. This is a very fast reaction and will happen anywhere between 15 minutes, and the reagents are pretty stable, and I'll get to that in just a minute. So that's the detection mixture. You incubate it for 3 hours, and the signal is stable for almost 2 days. You just measure the TR-FRET. And we pretty much use the HTRF setting. So any instrumentation, Envision or BMG Labs, Ferrostar, everything has default HTRF settings, so you should be able to measure TR-FRET very easily. So one thing I do want to mention before moving on to this is when the ligand, which is SAH, that binds to the aptamer, it causes significant conformational change. And we do see that once SAH is bound, this complex is pretty stable, but it does take about some equilibration time for the 2 pieces to come together and recreate, and that's why it's about a 3-hour equilibration time for the signal to stabilize. However, if you're running it in kinetic mode, and if you have a standard curve, you can start monitoring the signal anywhere from 30 minutes onwards. And if you have a standard curve that's run at 30 minutes, you can go back and interpolate your data. So definitely that is possible. Okay, so moving on, so to evaluate our methyltransferase assay, we often use start with standard curves. Now, the purpose of standard curves is to mimic the enzyme reactions. So we start at a given SAM concentration. So in this graph, we started at 100 nanomolar SAM concentration, and this is considered to be 0% conversion because everything remains at 100 nanomolar SAM. And SAM is then decreased incrementally and is replaced with SA, such that when you add them together, you know, a combination of SAH plus SAM should add up to be 100 nanomolar. Now, these curves were both done, and this is an example of 100 nanomolar curve, and this is at 1 micromolar standard curve. And both these curves were done with sufficient replicates of 16 to allow calculation of Z prime values. Now, Z prime is a measurement of not only signal variability, but also dynamic range. So it reflects on the usability of the assay. Now, for a biochemical assay, typically NIH guidelines says we want a Z prime greater than 0.5, but you can see here in this table below with a 100 nanomolar standard curve at 10 nanomolar in the enzyme reaction or in 10 microliter reaction, you get a Z prime of 0.83. Now, that's really great considering in a 20 microliter reaction, you're measuring 5 nanomolar SAH, so it's extremely sensitive. So what it indicates is, A, you know, when you run a screen or when you do a big experiment, you'll get unambiguous results, you know, because your Z primes are really high. So that indicates a robust assay. And secondly, these results here with such low, such good Z prime and such good lower limit of detection, we just, we hit our main targets was to get the sensitivity of the assay down to about 10 nanomolar or even lower than that. So this is really great for some of the low km SAM methyltransferases. So based on what we heard from our customers, you know, we got what we were shooting for. And as far as I can tell, there is no other assay that can give you such robust results, you know, at the cost that we are going to offer this assay. So this performance is really good. So then we wanted to, so this is another example of how you can look at the assay. Here is just a titration of SAH, and I started the titration really high at 5 micromolar, as you can see all the way down. And this is in 10 microliter, and when you see in the table SAH at detection, that's at 2.5 micromolar, so in 20 microliter volume. Again, the lower limit of detection was calculated just by titrating SAH alone without any SAM in there. And about 1.22 nanomolar of SAH gave us the lower limit of detection, which is defined as the first positive Z prime that we measure. And when you look at it in the final volume, it comes to about 0.6 nanomolar. So 1 nanomolar in your enzyme reaction, 0.5, 0.6 nanomolar in the final volume. And this is the EC50, about 10 to 12 nanomolar consistently that you get with the assay. Now, as I said, the assay is definitely, you have to pay attention to the most important part is the temperature. So if you want to get a very good sensitive assay, I would say incubate the plate. I mean, you can run the enzyme reaction at whatever temperature you want, but cool down the plate to room temperature before adding the detection reagents. That's really important to get a very sensitive assay. Now, this is some comparative specs that we did, some kind of benchmarking that we did with the competitors available. So we did the lower limit of detection, and that was just by titrating SAH as I showed, and in the enzyme reaction, in the 10 microliter reaction, it's about 1 nanomolar for our assay compared to about 30 nanomolar for the competitors. Then we did some EC50 titration. Of course, I picked PRMT4, which has Km, at least the apparent Km is, the literature values are about 130 to 140 nanomolar. So when we ran it at the Km, it was some of the assays did not work well, so we went higher to evaluate to have it as a fair ground for all the competitor assays. So this EC50 was determined at 2 micromolar SAM, and we found that the EC50 was 0.9 nanomolar for our assay compared to 10 and 33 nanomolar for the competitor assays. So the amount of enzyme that you would need for an AptaFluor assay is several fold lower than the other assays out there. Number of additions with the AptaFluor assay and the competitor one was 2. For some other assays, it was 3, which may or may not be a problem for people who have automation capabilities, but that's just an observation. Detection format was TR-FRET or luminescence, and the one thing I would definitely say is this is a direct assay, and there are no coupled enzymes involved. So as SAH is being formed, they start binding to the 2 pieces if you run it in a kinetic mode, and if not, you just quench the reaction and the SAH binds to the 2 pieces. We also determined the Z prime with PRMT4 again at higher SAM concentration just to offer a fair ground, and you can see that the Z prime, even though good with the other assays, we had a Z prime of about 0.8. Now this table below just gives again the Z prime that we observed with just titrating SAH with the 2 other assay formats, just to give you an idea about the comparison between the 3 formats. So the next thing we wanted to do was to take a hard look at the stability of the reagents and also the signal, primarily because it's an RNA-based sensor. So when we first started working with this Ribose Switch, we were really worried too, because it was RNA, and RNA has it's known to, you know, at least that's the stigma out there that it can get degraded. You need to either avoid RNases and use treat reagents with DEPC. And even though we started doing all that, we realized as we kept working that all we had to do was use nanopure water, nuclease-free water, and that's the only precautionary thing that I use to work with RNA. I have found that these RNA pieces are extremely stable, and probably once they form the secondary structure, they become really stable and they cannot be degraded. Anyways, so what we did, so this is the conjugation mix stability, and this is what I was talking about. So when you buy reagents from us, there will be this piece 1 and piece 2, and for the piece 1, which is just biotinlated, you would have to add the terbium. So we do mention in our technical manual that you have to make it fresh right before the assay. But here is a comparison where we made it 6 hours or 24 hours prior, or even 48 hours prior, and you can see that it is the 48 hours drops down a little bit, but even so, the Z prime values are really good, and we would really want you to do it right before the experiment, but this just gives you an idea that, you know, these reagents are really rugged. Now this is DEX stability. So what DEX stability means is once it's conjugated with terbium, you can mix the 2 pieces together. So you're adding piece 1, which is now terbium conjugated, and then piece 2, which has dilite. You mix those 2 together, and you can leave it in your bench. And all this was done at room temperature, so you can leave it on your bench. Like this was made fresh right before I had to add. This was made 2 hours, 6 hours, or even overnight, and you can see that once conjugated, these reagents are really, really stable. So this goes on to prove that the reagents are stable. Now, signal stability, we do say that it takes 3 hours, even though I mentioned you can chart seeing if your reaction worked or not. If you're running a kinetic assay as soon as 30 minutes, you would start seeing signal, but the signal does stabilize only after 3 hours. But once it stabilizes, it's pretty stable for up to 24 hours. So you can come back the next day and read the plate, and it's still pretty solid. So that's the signal stability. Now, since this is the first time we are launching an aptamer-based assay, and actually, I'm not sure, maybe the first time an aptamer-based assay is commercialized, I think. I don't know about that, but we wanted to take a really good look at the tolerance of all the reagents, and we tested, I just put a couple reagents here, but we have tested a whole bunch of reagents with the detection reagents to see how stable or how tolerant it was. And one thing that would jump out is, EDTA and EGTA are not The tolerance is only about 1 millimolar. And our speculation is that for the two pieces to come together, you do need magnesium chloride. And I think EDTA and EGTA chelate the divalent ions and thereby prevents the two pieces to come together to reconstruct the intact aptamer. That probably is the reason, and one way to get around it probably would be to increase the magnesium concentration. But other than that, the DMF concentration was less tolerant. The rest of the reagents were pretty well tolerated. Zinc was another reagent that was not tolerated at all, and I just show zinc acetate, but this we were curious about it, so we tried zinc chloride and zinc sulfate, and whenever zinc was involved, there was some problems. So thankfully, though, most of the methyltransferases do not require zinc. So this should not impose a problem to any of our customers because zinc is not used in the buffers for methyltransferases. So next, here, this is just an example of running it at a really low Km. So here, this is SET79 enzyme, and we got the enzyme from reaction biology. The substrate that was used is histone protein, H3 full-length protein. And the SAM concentration was 100 nanomolar in the enzyme reaction, so 50 nanomolar at detection. So you can see that I ran the reaction for 2 hours at 30 degrees Celsius. I could have ran longer to get a better EC50, but this just gives you an idea that you can really go lower in SAM concentration and still get a good assay window. Now, the assay itself is nonlinear when you plot the ratios against the enzyme concentration. And if you want to go back and look at how much SAH are you actually producing, you will have to run a standard curve under similar conditions. So what I mean here is we ran a standard curve with different concentrations of so SAM starting at 100 nanomolar, which would represent 0% conversion, and then decrease it incrementally while increasing SAH. But all this was done in presence of the histone protein, so you're mimicking the enzyme reactions, and then we use that to interpolate the data. So you can go back and exactly find out how much SAH is produced when you use about 50 nanomolar or 25 nanomolar SET7. Next, we wanted to see if typical methyltransferase acceptor substrates would interfere with the AptaFluor detection reagents. And this was of great concern to us because histones are known to bind to nucleic acids. So we were kind of almost positive that the RNA aptamer is going to bind them and it's not going to work for us. But and also nucleosomes. Nucleosomes is almost like a dirty prep, I would say, of histones and DNA together. So we were concerned how these reagents would work. It's like a crude fraction, and maybe it contains RNAs. So we were concerned about that. However, we found that the assay could accommodate any of these used substrates listed here, nucleosomes, histone, peptide, or poly-DIC. These were all tested, and by running a standard curve, and you can see right on top of each other. The only thing that I would like to mention here is if you're running it with nucleosomes or histone, we do suggest that it is important, and you have to stop and quench the reaction. So you break down the histone and then add the detection reagents. So that's important when it comes to these two substrates. If it's peptide or poly-DIC, you can run it in real time. We have not seen any problems with that. So you don't need a stop reagent in that case. And the graph and the table below actually conveys the same, in almost the same information. This shows the SAM concentration that we tested them at initially, and also the acceptor protein that was used, and the EC80 concentration of the enzyme. Of course, it's a nanogram per microliter, so it may not give you a lot of idea about nanomolarity, but I will get to that in just a bit. I'll show you some additional examples. So can this assay be used to determine enzymology like KM and Vmax? And absolutely, you can do that. And we have done that with G9A, with PRMT4 and PRMT1. I'm just showing one as an example here, where we determined the KM for SAM using G9A enzyme. And the KM was about 500 nanomolar or 0.5 micromolar, which is what the reported literature values are. So enzymology can easily be done using these assays. So next we took a closer look at some histone methyltransferases. The first one that I'm just going to share some data here. The first example is G9A-GLP. It's a complex protein, and it was titrated using 10 micromolar H3 peptide. The peptide was from 1 to 21 1 to 21 peptide, and I think we got it from AnaSpec. The SAM concentration was 200 nanomolar in all for this enzyme. So you can see that this is the complete reaction indicates SAM plus the peptide, the H3 peptide. The square indicates no peptide, and then this is just peptide by itself and no SAM. So we do see that this is very characteristic of many methyltransferases and kinases. So people who have worked with methyltransferases would have seen graphs like this, that you would see at high concentrations of the protein, you would see some kind of non-specific reaction. Either water goes into the pocket, or it's a common phenomenon seen with both kinases and methyltransferases. However, the concentration that you would pick for your dose response curves or to do hit analysis would be somewhere in this, would be in this region. And at that concentration, if you pick that, the contribution due to just SAM alone or peptide alone is very little, as you can see in this graph here, where the ratios were converted into the product formed, which is SAH, using a standard curve, which has peptide in this case, to mimic the enzyme reaction. So you can convert that into product formed, and you would see that at the concentration that you want to use, about 1 to 1.5, right in this region, there is very little contribution, non-specific contribution due to the controls. But when you do the titration, this is a typical example that you would see. Now, this is an example where we did it in kinetic mode, where you can stop where it's not stopped, it just, the reaction keeps progressing, and you can run it at different concentrations. And you can do this because it uses peptide as the substrate. So that just gives you an example of things that you can do with this assay. And we determined the Z prime, which would indicate, what kind of assay window you would get with this assay. So you can look at this particular example, and with both sort of controls, with just SAM alone or just peptide alone, you still get a Z prime of about 0.8. And this was done using 1 nanogram per microliter of the complex protein that we saw. Now, what this reflects is the Z prime values about 0.8 reflects the low standard deviations that you get with AptaFluor assay. It's about 1.5% for the positive controls and less than 5% for both the negative controls. Now, these statistics reflect the stability of our reagents, because if there was any degradation of the RNA aptamer, we would see that immediately with the signal variability. And if there was SAH in any of these controls, the assay window will start dropping over time. So you would identify that right away. Now, this example here just shows the dose response curves for 2 specific G9A-GLP inhibitors. And we use for these kind of experiments, you would use the EC80 from the previous enzyme titration, and then IC50 was determined, and we convert that into % activity to report the IC50 values. Now, right beneath the 2 compounds, this is the UNC compound and the BI compound, and right beneath it gives the literature values. So it's slightly twofold off for the UNC compound, but the BI compound is right on top of what was reported in the literature. So another example, I just wanted to show you some additional examples. This is PRMT5, and in this you don't see any kind of response in the absence of GST GAR or SAM alone. So this is a very clean prep. And then when you convert it based on the standard curve shown here, you can see the linearity, the initial velocity condition. So that's good to know. And this was run at 200 nanomolar. So this right about here represents 10% conversion. 20 nanomolar represents 10% conversion, which is at initial velocity. So this PRMT4 is another example which uses histone, full-length histone protein, and a titration is seen here again at 200 nanomolar SAM. The KM is about 140. For PRMT5, it was about 150. So we picked 200 nanomolar to run all these experiments. Again, as I said, the ratio themselves would be nonlinear, but just by running a standard curve, you can convert them into a beautiful linear graph. So again, with histone, it's working with histone is not very easy either. It's a full-length protein, so we wanted to see if the Z prime gets affected because we use, I mean, earlier we had used peptides, which are easier to work with. So this is an example where we use histone protein and still get a very good Z prime of 0.8. And then we tested DOT1L with nucleosomes. So as you can see, with nucleosomes, working with nucleosomes, people who have worked with nucleosomes would agree with me a lot. It's different from lot to lot, and we do not make our nucleosomes in-house. We purchase them from reaction biology, and there is always that lot to lot variation. We have to qualify each lot, but it works really great with the AptaFluor assay. Of course, you would see, like I mentioned, you do see in the with just nucleosomes alone, you do see some kind of response, but the concentration that you really need to use, about 1 to 2 nanograms, you don't see much happening there. And then we use that concentration, 3.8 nanogram per microliter, to run the dose response of 2 known compounds. These are epizyme compounds, and the IC50 values are shown here, which I think the reported ones were even more lower than what we see, but this is what we obtained using our assay. So the last thing I want to share today was we took the Cayman Epigenetics library. It's about, I think, 98 compounds, like, if I can remember that right. So first we ran it through just our detection reagents, which is the 2 pieces of RNA. And of course, SAH was one of the compounds in their library, and cinefungin, which SAH and cinefungin are very similar in structure. So SAH, cinefungin, and SAM share structural similarity. So it came as no surprise that both these molecules caused an increase in the ratio. So that, I mean, of course, this is an SAH assay, so this was no surprise at all, and neither was cinefungin. But this compound, AG014699, caused a decrease in ratio. So this is like an interference assay, and here are all the controls. And then we ran the screen through G9A enzyme with the G9A enzyme. And just for reference, I have just the detection reagents up there. So what you can see is, yeah, some of these compounds, of course, cause an increase in ratio, but we did see, we could pick out like some of the hits here, and these, the ones in the rectangle, are validated. We have IC50 values for them, but we did see a couple other compounds that showed up. So we are looking into that. So that's where we are at, and the conclusions, the summary of today's webinar, I would say it's a unique assay. It's different. It's one of its kind. It's aptamer based, and it has really good selectivity and affinity, and it's from a naturally occurring ribose switch, and enables direct detection of SAH with TR-FRET signal, which is really popular among a lot of pharma companies. The lower limit of detection is about 1 nanomolar SAH in the enzyme reaction, produces outstanding Z prime values, about 0.9 at 10 nanomolar. So that's really, really great. Very good reagent and signal stability. There is there is this thinking that, RNA is difficult to work with, RNA might not be stable, but contrary to all that belief, the reagents and signal were really stable, not just for hours, for days actually. And that provides you flexibility for taking a long lunch break or coming back the next day and reading your plate. So it's really useful that way. And the assay is universal. It tolerates most of the common acceptor substrates to date. To this date, we have tested histones, peptides, nucleosomes, poly-DIC substrate, DNA. It's worked really well with all these substrates. So it's a different way of testing some to get very sensitive response. I will take questions now, and you can type them, as I said, in your question box. And if you want to ponder over your questions and send us an email, that's fine too. My email address is there, and I'd be happy to answer any additional questions. And if you want to give this assay a try, which I would highly, highly recommend, just give it a try. It'd be fun working with an aptamer. So you can place an order either using this email or call us up. That's the phone number. And I think we will have these slide deck probably available to you, so you can listen to the presentation. And if you have any questions then when you listen to it again, you feel free to contact us. We are happy to answer any questions or concerns or anything that you might have. So with